Hai, Assalamualaikum warahmatullahi wabarakatuh. I am a representative of Group 3 to represent our research proposal, which is Chapter 1 Introduction. So here is our group members. We'll go to the next slide. Okay, 1.2, we continue from background of study. So the next is problem statement, which is the difference on spawning efficiency of beta splendens. So in aquarium specimens of Siamese fighting fish that readily accept processed dry fish food as well as large invertebrates show that they are omnivore. This confirmed by Etherod et al. At 1971, Sterba 1983, and Sakurai et al. At 1992. So they eat a different food. Normally, these fish food feed on plants above the water, but when they are hungry, they eat tiny benthic fish. This fish feeds on zooplankton, larvae, and insect ova. This is confirmed by Argini in 2000. So as a result, it will introduce the finest feeding treatment and how the kind of treatment affects the timing of spawning of Siamese fighting fish, as well as the amount of ova and larva. Different foods on spawning efficiency of Siamese fighting fish. Important to get fundamental understanding about the possibilities of increasing the amount of spawning of brood fighting fish and the most larva generation. This research by Sahabayokani et al. At 2014. Many studies on broadcast spawning marine invertebrates give gonadosomatic index values, which are typically highest around the seasonal peak of breeding, when extreme values of up to 75% and have been recorded. This is confirmed by Ciavereno, Menzin, and Ramirez at 2004. The mature gametes appear to make up a high proportion of gonad mass at peak breeding times. Many different gonadosomatic index have been used to assess reproductive conditions. The research was by Lawrence and Lane at 1982 and Ebert, Hernandez, and Russell at 2011. So, many papers give separate male and female GSI values. Others test for sex difference and pull the data if no significant sex difference is found. Although these species have evolved excellent breeding methods, the high incidence of mortality at the time of larvae generation is a serious problem. Fish larvae are often physiologically undeveloped, exceedingly tiny, and fragile. This is research by Sri Krishman R. et al. 2016. Harris and Atapatu at 2012 reported that a combination of elements such as nutrition, substrate, water quality, pathogens, and stress determine a larvae chance of survival. These numerous studies on the feeding of larvae have shown that live feeds promotes higher stress, determine a larva's chance of survival than formulated feed or artificial feed. Since it guarantees great development and survival, Artemia has been widely employed in Sri Lankan fighting fish larvae, rearing as the only source of live feed. However, Artemia's high price and sporadic scarcity render it unsuitable for commercial aquaculture. This research by Lim et al. At 2003. So we go to the next, which is the significance of study. This study's research greatly benefits individuals, institutions, and agencies. Based on our title, the benefits of this research are to describe a new species of fighting fish called Beta cohene. This new species only occurs in the most northern region of Malaysia and southern parts of Peninsula Thailand. This study will also provide information about the characteristics of this fish based on its external and internal features. Because fecundity and gonadosomatic index play an important role in the development of today's fisheries. The finding of this study will benefit the community. The increased demand for fisheries graduates justifies the need for more effective research approach. As a result, institutions that use the recommended approach based on the findings of this study will be capable of effectively in training students. The gonadosomatic index and fertility are two of the most important parameters in studying each fish reproductive biology. Gonadosomatic index is a parameter used in the study of fish spawning biology. It also determines the degree of ovarian maturity. The gonadosomatic index is the ratio of gonad weight to body weight in fish, because the ovaries of gravid females increase rapidly just before spawning. The SI is especially useful in determining the day and season of spawning. This study will assist those involved in the research process in the identifying critical areas that the researcher may be unable to investigate. As an outcome, a novel approach to studying the factors and effects of fecundity and gonadosomatic index of beta-caenae can be developed. Furthermore, rain is a key component influencing the spawning of many tropical fishes. Many tropical researchers believe that the breeding is seasonal in many fishes. The influence of water hardness on oviparous and ovoviviparous fish biology in semi-natural aquaria by assessing adult and larval development, as well as reproductive performance in terms of gonadosomatic index, fecundity, fertility, hatchability, and larvae survival. Furthermore, the overview offered in this study will encourage the development of new paradigms that will be valuable for future discussions on the gonadosomatic index and the fecundity in beta-caenae, perhaps leading to a more in-depth examination of it. So this is our aims. This study proposed will investigate new mouse breeding species that has not previously been listed is described. So furthermore, we go to the specific objective. So the specific objective for this study is to determine the breeding performance of beta-caenae and also to conserve beta-caenae in its wild habitat. So we go to this is our reference that we will use in our study. So that's all from me. Thank you.